To generally stay on track, I'll just give Bjorn a minute to catch his breath. But we will move into the second presentation of the day. So it's the counterpart to the T cell class 1. It is the T cell class 2 tools and pipelines. And Bjorn will be presenting that. So thank you so much, Bjorn.
So T cell class 2. The difference of class 2 versus class 1 is primarily that MHC class 2 molecules are not present on all cells, but on professional antigen presenting cells and actually many normal cells when they get immune activated through inflammatory signals can express MHC class 2. And so these molecules present peptides that are derived from extracellular proteins primarily, where they can be taken up by the cell, processed through the endosome, lysosome, and trimmed as they bind to MHC class 2 and then be presented on the surface. So these can be often things like bacteria, secreted proteins, debris from other cells. So say, for example, in the context of cancer, you can actually have presentation of cancer cells that get killed, and then you have via class 2 presentation of the debris from the dead cells to other CD4 T cells. And CD4 T cells are, in contrast to class 1, which are recognized by CD8 T cells, what recognizes MHC class 2 restricted molecules. So for class 2, you have two chains. For class 1, you have the alpha chain, which is polymorphic, and the beta chain, which is monomorphic. For class 2, you have potentially two polymorphic chains, alpha and beta, that form a complex. And for both the DP and the DQ molecules, which consist of a polymorphic alpha and beta chains, for DR, which is the one most commonly studied, you actually only have the beta chain being polymorphic and the alpha chain being monomorphic. There's also DRB345, so you have DRB1, which typically people talk about, and then you have a linked locus, which people only have one copy of it. They either have DRB3 or 4 or 5, and those are inherited together with certain alleles of DRB1. So this is one of the actually interesting issues. This is one of the more interesting MHC class 2 molecules, and it's often ignored because the structural variation in the genome is kind of hard to capture for a bunch of tools. And it's often not typed, but it's just as important. And it's not typed because you know actually which DRB3, 4, 5 you have based on the DRB1 allele you express. So the key difference for class 2 versus class 1 molecules is that there is this binding core, and I kind of showed this in the previous slide, that actually makes contact to the MHC class 2 molecule. And then there's residues that are hanging out on the sides. So this means you can have peptides that, if you would put them side by side, like align them directly, would look like they have nothing in common. But if they share the same binding core, peptides that look very different might bind the same molecule. And this leads to some of the issues here with identifying binding motifs and training algorithms. Because you can't, in contrast to class 1, where it's really easy actually to identify motifs. For class 2, you have to do some extra legwork to find the binding core first. So you find the tool just as class 1. On the main homepage, you click on class 2, and then you see a page that now should look very familiar, where you have, again, we have this example sequence pasted by default, and you can obviously add your own here. You can change the prediction parameters like peptide length. We generally work with 15 mers, which is the minimum length you need to get a peptide that works with class 2 is typically 13. We end up using 15 in our experiments, which is a nice compromise. You can go longer, but then synthesis becomes more expensive. And the problem, though, is like if you now synthesize 15 mers that overlap your input sequence, and they overlap all of these by one. As I told you before, there is this shared peptide binding core. So many of these peptides are going to be predicted, if this is a good binding core, many of these peptides are going to be predicted as being great binders while they're essentially the same region. And actually, if you do ligand dilution, you'll likely find many of these and extensions thereof, many different variants of the peptide that share the same kind of like ladder centered around a shared binding core. So in the prediction, we don't want to represent all these different variants that are essentially the same underlying reason for binding. So one of the things we have been doing and using ourselves in the lab is to actually shift peptides. And what I mean by that is you take the original sequence and you don't make every 50 mer, but you shift them by 5 amino acids, and that shift is what you can do. And then your the 9 mer cores that you have are represented um, not like 7 times in these overlapping peptides, but there's some overlap that allows you for some leeway in the directly flanking residues, but overall you're reducing the otherwise redundancy that you're getting from the sequences that you have. And that's what we recommend to get rid of this um, redundancy. Then we have the MHC class 2 allele selection. This is again the exact same kind of thing like what Jay was showing for class 1. When you click on the finder, you get this kind of menu and you can start entering in here. As you start typing, it'll start auto-completing. So you can start typing in here and you should find the alleles that you're interested in. Then similar to what Jay was showing, there are predefined sets of alleles. There is a 27 allele set hidden here conveniently, which is representing the most common MHC class 2 alleles. And then there's the 7 allele panel, and I wanted to talk a little bit more about this. So this 7 allele panel is based on the 7 allele method. What we were trying to find out is like what sets of alleles are driving or can be used to best predict peptides recognized in the general population. So we took data sets where there was an unbiased selection of peptides overlapping 15 mers, and they were all tested in a large panel of individuals, and there was like responsive and non-responsive, and then we tried to like narrow down what predicted which peptides were responsive and not. And here what we ultimately found is that if we predict each peptide for its affinity for 7 different alleles, and take the median of that, that gave us a good predictor of the population response. I have to stress here, population response. So if you know the specific alleles of an individual donor that you want to get a response for, obviously input those. If you have no idea, this set of 7 alleles is a good one to give you an idea of which peptides are going to be likely having recognition in a broad population. So if you select that 7 allele panel, that's how your website will then look like. The 7s are going to be filled in. And now you can select your prediction method. And this is, again, very, very similar to what Jay showed for class 1. You can select your methods here. And we have this recommended thing, which is based on, again, benchmarking. So we are taking benchmarks in weekly for MHC class 2. Like every week, we have new releases of data in the IEDB, and then we are running this data through the prediction methods. Actually, there isn't that much. There is not every week a new data set for MHC class 2 binding data available in the IEDB. So as you see here in the dates, this is when new data sets become available, and they need to follow certain minimal criteria, meaning a minimal number of peptides. They need to have clearly defined restriction and these kinds of things. And then we include this in the benchmark, and we found that the BA method, which predicts binding affinity, is best at predicting binding affinity, unsurprisingly. At the same time, and I'm not showing this here, in our separate benchmarks, both of MHC ligand elution data and of actual epitopes, using the EL output is better. So, and similar to what we see for class 1, the EL output, which essentially is predicting what peptides are not only binding, but also overall likely to be presented, that is a better predictor of T cell recognition, actually, as we would actually hope, um, than the binding affinity alone. But if you really care about binding, and there's many reasons why you'd want to do that, then the BA predictor is obviously better. So anyway, by default, we always stick to the standard recommendations here, and we are somewhat conservative in changing these over time. We don't want to change our recommendations every few months or so. So we're kind of waiting until there's like clear and convincing evidence before we switch. So you can add other prediction methods here, similar to what Jay showed you with adding processing or immunogenicity here. But I'm going to not do that. I'm just going to run this. And then I'm going to get a feedback like this, where the peptides are reiterated, the start end positions, etc., each of the alleles. But importantly, you have this median binding percentile. This is now the median of the 7 alleles considered that is being repeated for each peptide. And you have these different tables. Sorry, I should have pointed this out. So here, you're on the peptide table output, and switching to the consolidated peptide table output, you have these here. Given that you only have one prediction method running, these two peptide columns are redundant, and this is the one that actually they have the same values. This is the one you'd want to use, and you can now use that in principle to select peptides that might have the highest likelihood of being recognized in the general population in this protein. Processing. So class 2 processing, again, like you have this protein being taken in, and there's this trimming steps. There's lots of trimming steps, and there's binding of the core to MHC class 2. So as I mentioned before, actually, when we elute peptides, we actually often see different variants with different levels of trimming being presented, but there are motifs that are driving where this cleavage happens more so than other positions. So, for example, there's a quite typical stop motif. If you have a proline in position 2 here, you'll likely have a cleavage here and not one further, for example. So we have been using MHC ligand elution data and identifying these cleavage motifs at both the C and the N terminal of peptides. And then we have been using that to predict which peptides are naturally processed and presented. And we see a marked improvement when combining the binding and the cleavage motif to identify the MHC ligand. However, and I'm going to put this up top because otherwise it would be so much of a downer. We were so happy with this, but to predict T cell epitopes, we do not get a significant improvement of the prediction quality by knowing the exact ligand being recognized. And I'll tell you why in a bit. So here is the tool. This is the MHC2 NP tool. So you could have selected this down here from the prediction model. You have prediction parameters as before. When you run this using this run button, you are getting again like these. You get an NP motif. Sorry, an N terminal motif, a C terminal motif, and a score based on the peptide length, which overall gives you a cleavage probability. So for a given peptide is being judged by, again, like the length, the N and C terminal cleavage motif, and then the higher the score is, the higher is the probability, and the percentile ranks are as always normalized based on the score distribution to make them easier to compare. And when we now look at this, at the score, if we use the binding alone, sorry, we are combining the binding score and the cleavage score here in this paper, and then we get a better recovery and better prediction of eluted ligands by combining these together. And so there's a certain weighting thing that we find to be optimal to get them back. Combining actually the 7 allele method that I talked about before with this binding score. And then again, like if you really want to know what peptide is naturally processed and presented, this is a great way of identifying the specific peptide. Now, we worked on this for several years. Morton's group has also worked on this for several years, and we tried to make this into a better predictor of epitopes and neither group found that we could. And the reason for that is actually if you have a T cell recognizing your perfectly exact processed and presented peptide, and now you add a residue to it or you remove one, the T cell will still recognize it just as well. Knowing the exactly correct ligand is not important for T cell recognition because the T cell recognizes the core close to where the MHC binding happens and what flanking residues are flopping around in the on the outside is not that important to get correct. And in reality, when you look at MHC class 2 peptides that are being tested for T cell recognition, typically people do not pay attention to the terminal residues and extend them to 20 MERS or 50 MERS or whatever is convenient for synthesis. So with that, I'm summarizing kind of our thresholds for MHC binding prediction of strategies for MHC2 binding predictions. So I do want to point this out because people are still talking a lot about this, that MHC2 predictions are worse than class 1. We don't see that much anymore. When we look at the ability to predict binding or MHC ligands using the current prediction methods, they're very comparable between class 1 and class 2. In the past, we had been recommending things like the percentile rank less than 10% rather than 1% for class 2, and an MHC binding affinity threshold of less than 1,000 nanomolar, then less than 500 nanomolar. And if you're caring about MHC ligand predictions or binding predictions, you can actually now be much more stringent. And we know that the titer cuff of like a 1% percentile rank actually works just fine for class 2 peptides when you're trying to predict natural eluted ligands for that specific allele. However, whenever we look at T cell epitope recognition, where it has to be said that in many cases the restriction of the class 2 epitope is often not clear. While the EL scores outperform BA scores, so we totally go with that, we still see that there is more looseness in what is being recognized by T cells. And we have multiple, I have a wet lab and a dry lab. In the wet lab, we've been trying and trying to improve on this and get the best selection method out there. And we are still having to be more loose for the MHC class 2 binding than we would like to be. And this is if you want to have a project to work on and find out what the explanation for this is, please do a comprehensive evaluation of the factors. There must be other factors that are highly impacting this is outstanding. Obviously, we have our list of preferred candidates, but this is not something we can say is solved or settled. So I wanted to briefly talk about class 2 pipelines and give another example here for class 2, how we can use pipelines to do more than just definition of affinity. And I am going to give an example where we take predictions of likely ligands, then using PepMatch to find homologs of those in human proteins, and then look for expression of those peptides in the lung using PepX. So here's the how that looks like, and I'm going to do now what I should never do, namely do a live demo. So if we are here, this is what you saw before. This is now the peptide. It was already run for 7 allele method, run on the example sequence, which is like the SARS-2 spike glycoprotein. We did like the 15 MERS with a 5 residue shift, 7 alleles. Now we have these peptides. And one of the questions that came up in the context of SARS infections is like, are there potential residues in here that might be similar to humans? And that could be leading to autoimmune reactions. So the question then here is we have these peptides, and first of all, okay, let's say we want to have the median percentile has to be less than 10. So we can do that live here. And so now we have just 185 residues left. And I can take these and now add the next prediction step, which in this case is pep match. And I am going to use um, I'm going to use the core. So you can choose what are you going to pass on. This is the core. I could also pass on the original peptide sequence. That's these, right? And then I can see here now pep match is here. It's going to take that column which is highlighted, and I can say I want to have all the peptides in the human proteome. They have three mismatches to these peptides that were from SARS spike protein, and ask, do these exist? And I run this, and I'm not going to wait for the run because, as Jay was saying, we have issues here with multiple people using the site. There's warnings popping up which are pretty much saying we have the same peptide repeated over and over, which will happen if you run these predictions for multiple alleles. So there's not really a real warning. And I'm just going to go and jump to how the final result looks like to avoid the time here. So we have, in this case, here in this example, and you can actually follow this if you have the slide link. This is again like a pep line you can directly get at in the pep, right? You can copy these as Jay was showing. It takes the core sequences that I predict. It looks for matches with one mismatch, one or up to three mismatches in the human protein. You see these things coming up here, and then you look for expression in the lung of these things, and then you actually have here an interesting peptide found in a similar, a peptide in myosin, which is actually has been implicated. Like there has been um, concern about myosin cross-reactivity of after SARS infection, and you can now say, well, not you have a similar peptide in human to the SARS peptide, and it is expressed in the lung. However, one has to point out here that this has uh, um, a mismatch in a 9 residue stretch. So we did actually a bunch of analysis showing that this is not an unexpected level of matching that you're finding. If you look at the whole peptide sequence, for example, you will actually not find any great hits. But that sorry, I'm switching here between biology and the pipelines. I hope you see the point that you can select peptides from one tool to pipe into the other and to the next. And in each of these steps, you're going to get additional information that you can then combine to uh, guide your future work. Okay, that was the pipeline, the API. I'm going to show like a quick movie here. Actually, I showed you this before. You can copy the API post request here, go into the API tools overall information page and scroll down to the pipeline section. There you can now paste in whatever you have, right? You can design on your website the pipeline, put in here what you have, and run it directly and see you're essentially running exactly the same query that our front end is running against our back end, and you're going to get your result back that you can then modify. So, I mean, this is a way essentially how you can learn how to use the pipeline. Here is like the JSON format of the input to the tool, and then there's like the output here. We would recommend, if you want to start working with the API, just use the website to design your kind of API calls, and then modify the code. You can have direct access to the code. You can try it yourself. So this is, you get started on the page. You can search for tools on the all tool list. You have the API. I just showed this, and you can run through them. You have the help documentation available, and you have the download of tools available for the next gen site specifically. I did want to point out overall the downloads of tools is, as I mentioned yesterday, often important for people who want to not work outside of their firewall with the sequences that they are working on in a proprietary fashion. So please, if you're a commercial user of these kinds of tools, consider licensing the tools. Um, there's a contact email here, and there's been a lot of patents coming out of people that people have generated using IEDB tools, and there's a lot of active licenses from pretty much every big company working in this field. Has been using them. I think they're very reasonably priced, and they allow us to provide support for these standalone tools um, and answer questions, etc. Obviously, if you're taking the licenses, we will essentially we have one FTE paid for by the license income that they're getting, so that that person can help with any problems that you have. So with that, I think I'm on my time, but we're still over,
Thank
um, and
you.
I'm done. Yeah.
I will give you a few minutes to take a look at the chat because we still have some of our other questions and a few that have come through for the new presentation as well. And then I will also like to invite Jay back to the virtual stage to help conduct the section 2 Q&A. So Jay, do you want to take one or two to give
Or yeah,
Bjorn?
I'll take one of the more recent questions related to class 2. Ah, that keeps moving. All right. This question comes from Zudiz Zanzak. Hi, I could not find the IAG7, excuse me, mouse MHC allele in the class 2 T cell prediction. Is there a way to add this allele manually, or is it not supported? Um, so if, and I'm not familiar with this particular allele, but if it's not available through that autocomplete interface, that just likely means that NetMHC 2-pound doesn't have any training data for it, and it's not supported. Now, NetMHC well, NetMHC PAN allows you, if you know the sequence of the MHC molecule, NetMHC PAN allows you to input the pseudo sequence for that molecule. I cannot recall right now if NMHC2 PAN allows that, but I'll find out. But neither of those options are available through the next gen interface. You would need to use the downloadable tools to do that.
Thanks, Jay. I'll take the next one. Anonymous. Shouldn't we consider also the TCR recognition in the prediction process? Is it something that could be implemented, or could you explain why not? So first of all, the immunogenicity tool that Jay was showing is implicitly capturing what peptides are likely to have a TCR repertoire that recognize them. And so this is the problem. When you're asking for TCR recognition, you're not asking a well-defined question. You're asking, does this peptide MHC complex, is there a TCR receptor possible to be generated that can recognize this? And what is the likelihood of it being generated? There's not one TCR, there's billions. So what we found in the class 1 immunogenicity score is that certain residues that are likely to stick out more of the groove, that's at least how we've been interpreting it, and more likely to make contacts can be driving this. And for class 1, this has worked. For class 2, we have a tool, but we have not actually seen that it reliably predicts immunogenicity, and that's essentially what TCR recognition is, immunogenicity. And I hope I answered at least part of this. Happy to follow up more. Jay?
Yeah, and kind of a related question from anonymous attendee: how to predict, and this is going back to class 1, I believe, how to predict the peptides using the immunogenicity parameters, and what is the threshold value to be taken. So the immunogenicity tool outputs a score from -1 to 1. Lower is less immunogenic, higher is more immunogenic. The threshold that we had established in the paper, I believe, was about 0.2, 0.16, somewhere in that range, but I'll find that and type it in the chat.
Okay, next one is from Amit. With peptides with common core sequences, depending on starting position of core sequence in this peptide set, the affinity and Lewis score changes. With the 5 amino acid shift method, how are the values assigned to the core sequence? So essentially, the first step of the prediction method is pretty much to find within the longer peptide that you have is the core. And the vast majority of the score of your longer peptide is going to be driven by that core. Yes, there are some slight changes of where that core is positioned and what the exact flanking residues next to it are, but the impact is relatively minor. So by doing the 5 residue shift, you are missing some. If you want to, please insert all of them. You're just going to get 5 times more peptides, and then you have to do a post prediction filtering step. And you might want to choose always the best peptide that comes out of these based on the scores. But we consider those differences quite minimal, and it ends up causing a lot more issues of later removing these peptides also, specifically if you're working with sequence input sets from related sequences that have some mutations in there. It gets really complicated. So this filtering in the process that we're using results in a lot less data to handle, and we have not really seen in practice a real difference. Jay?
So from Mauricio Calvo Calle, for the peptide homology, would it be possible to restrict the homology to a given position in the peptide sequence instead of just a given number of residues in the sequence? And I'm assuming this is in relation to the PepMatch tool. I don't believe that's possible. I think the only parameter we have is overall identity.
Next question is from Tim, University of Edinburgh. Given the complexity of DP-DQ with both alpha and beta chains being polymorphic and the opportunity for interhaplotype sharing, how useful are DP-DQ predictions compared to DR? So this is actually similar to the old concern that we've shared for the longest time of being class 2 being worse than class 1. With the current state of data, we actually believe we now have a good handle of DP and DQ molecules in a similar degree to DR molecules. Yes, there's more complexity. Actually, one of the issues is also then when you have data sets from a given individual that you just get the typing of this is the alpha chains for DP, these are the beta chains for DQ, and you don't know which combination is important. There has been a great paper, and we should link it here somewhere. I mean, there has been previous work, I think is F.E. Petersdorf, and then now Morton had been computationally showing the same thing, that the pairing that actually is functional ends up being alpha and beta pairing that are co-inherited somewhere in the world. That doesn't mean that it is only just cis pairing of alpha and beta on the same chromosome, but essentially things that are never on the same chromosome tend to also not be functional. So there is actually less complexity in terms of like many of these combinations just don't get made when you look in more detail. And this is actually one of the things we wanted to include in the allele selector approaches. Jay.
Yeah. So next question from anonymous attendee. Based on my experience working with the tool so far, it seems that we can't enter, for example, 20 BOLA or HLA alleles into the allele section all at once, and adding them one by one is time consuming. Is there any solution or feature that allows us to import multiple alleles together at the same time? Sort of. So we don't have that currently built directly into the web interface. But you might notice on the web interface, when you make your allele selections, there's a little down arrow that says download prediction parameters. So you can download those prediction parameters as JSON, and then in there you can edit the list of alleles. We don't currently have a way of re-uploading that into the interface, but you can use that in the API, or you could use that to drive the standalone. But we don't have that built into the front end, although that is a pretty commonly requested feature, so we should prioritize it. Bjorn.
Next question from Anonymous is how to interpret the PEP-match data. Does 100% homology indicate likelihood of immune tolerance, while mismatches may indicate an increased risk for cross-reactivity? So yes, and we actually have, and we should put it in the guidance somewhere. The best study that we've had so far is the cross-reactivity for milk allergy. That's actually been a fantastic study in my mind, where we looked for which peptides in cow milk are tolerated by humans and which peptides are not. And essentially anything that has 0 mismatches or 1 mismatch is tolerated. So cow versus if you have 2 and 3 or more, so essentially all the ones that are nearly all the ones that are reactive had 3 or more mismatches. So that would be our this would be my personal threshold. At the moment, you have 3 or more mismatches between 2 peptides that are 15 residues long in class 2. Your chances are high that there's going to be no tolerance for these peptides, and you are likely to potentially have cross-reactivity. Jay?
And so actually might need some help from Bjorn answering this one, but this is from Honey. As I studied the IEDB's guidance, it states that the proteasome and tap processing predictions for identifying MHC1 epitopes are available only for humans. How can we apply this approach to non-human species such as cattle? And so, yeah, the proteasomal cleavage predictions were based on data, in vitro data from a human data set. And I think we have a warning on the site that if you're going to be using this for other species, just be aware of that. And I'm unfortunately not familiar with the how that translates into other species, but Bjorn might be able to add on to that.
Yeah, I mean, we did the comparison of mouse versus human, and there were clear differences. So there is some concern. In mouse, like the tap molecule has different specificity than the human one. There is no systematic comparison of these across different species. So I guess like use with caution, I think is the summary for that. And then I see Nina's coming on, but I think I'll do one more. For the benchmarking, do you combine both cleavage score and binding score? Is it just one cleavage method prediction? So the benchmarking that we have showed you so far is for binding predictions only. There is no cleavage involved. These are measured MHC binding values that we predict. We are actually in the process. We have a paper ready for actually doing MHC ligand benchmark, MHC ligand dilution benchmark predictions that involve cleavage. So, but what we've shown so far, it was all just binding. Thank you. That's it, I think, Nina.
Yes, thank you so much, Bjorn and Jay, both for the presentations, but answering a massive amount of questions. We still have a few, and I've taken about 5 minutes out of the break. So apologies, we'll just have a really short break now, but we'll do our best to get back to some more of these questions via a typed response. So for the attendees in the answered questions, that some of them may have a written response below them. But again, thank you, Jay. Thank you, Bjorn.